name is Jackson Dungan, and I'm a student at ASU majoring in biology with a focus on biological and ecological conservation. The title of this video is Rampant Evolution in Invasive Australian Cane Toads and was created for the Bio 182 video presentation final and will pertain to an invasive species that has taken over Australia's ecosystems. We were tasked with finding a notable and reputable media conglomerate that had an article discussing a topic of which is related to something discussed in lecture throughout the semester. The article I had chosen is called Cannibalistic Toads Reveal Evolution in Fast Motion Study Finds, published March 15, 2022, written by Annie Roth, published by the New York Times. Now, it was required that the article be a summary of a scientific primary literature that had been published no later than the start of 2019. The article I had chosen was called Choosy Cannibals, Targeted Consumption of Conspecific Hatchlings by Larval Cane Toads is Triggered by Species-Specific Defensive Toxins, published March 1st, 2022 by Michael R. Crossland et al., published by John Wiley & Sons Limited. To summarize this article, the main idea focuses on the cane toads, with the scientific name Rhinella marina, specifically in Australia, where it has become invasive since introduction in 1935. The focal point of the research behind the invasive cane toad is a toxin called bufadienolide, a toxin that enters surrounding water released from cane toad tadpole eggs after hatching that, when interacted with the amphibian, makes them one of Australia's most poisonous species. However, the importance behind this finding is the rampant evolution that took place in order for the cane toads to develop this trait to be consistently expressed throughout the population. The article also discusses the fact that because of the overcrowding populations of cane toads, an ecosystem capacity can only reach a certain limit before resources start becoming exhausted. The cane toads responded by becoming far more cannibalistic than previous generations had ever been. At this point, the primary scientific article now becomes relevant and is reviewed within the news story through direct quotes from experts in herpetology or other biologists who were either involved in the study, reviewed in the article, or were not involved in the study but still provide relevant information to further justified reasoning or feed additional information to the study or behavior of cane toads. The experiment was conducted in Australia's Northern Territory, specifically at the Adelaide River, where cane toads were collected for research. Cane toads were held in enclosures that simulate their natural environment and spawned eggs throughout the night after being injected with synthetic chemicals to induce a greater chance of spawning. 7 total clutches of tadpoles, quote, 4 tadpole clutches and 3 toad egg clutches, end quote, were precisely selected for the first experiment. Toxins were either used or withheld in the select experiment to determine the effects that the chemical had on the toads and if there was any correlation between the chemical and cannibalism. When tadpoles were present in the experiment, it was found that frog eggs were less likely to survive than when tadpole eggs were not present. However, what is more relevant to the hypothesis at hand is that when cane toad tadpoles were present, at the rate at which cannibalism occurs exponentially rises. What was concluded from the experiment was that, quote, comparison among the exposed treatments showed that the cane toad tadpoles consumed fewer eggs and or hatchlings of each native frog species compared to those of conspecifics. They found that the eggs were consumed by other toads prior to being independently swimming tadpoles. The data found showed that when the toxins were present, eggs in young tadpoles were 3.31 times more likely to be eaten by cannibalism than their counterparts of the toxin not being present. However, the discussion notes that the effects of the release of this toxin are likely to be more extreme in a natural element and not with a single controlled tadpole with artificial scenarios, but their data justifies the reasoning behind the hypothesis that toxins cause cane toads to become more cannibalistic against their young. The article follows an incredibly accurate path and is always able to give justifiable reason as to how each fact stated is true. Each fact that is stated within the summary can be found in the links that are provided in text after further research are backed up by outside research. The validity of the summary is solidified by quotes from professionals in the field of herpetology or biological sciences as a whole. By using such reputable and credible sources, all facts can be considered to be true. There is no additional work necessary to produce a more accurate summary, and all facts and statements are justified, meaning accuracy should be as close to 100% as one can possibly get. Carefully planned consideration was taken into account when gathering cane toads from the Adelaide River and were placed in artificial environments that simulate their natural environment in order to ensure the most natural reactions possible from the toads. Several different approaches were taken into account for every possible what-if scenario and to provide further justification for their data. In the last 2 tested experiments, a 2 milligram concentration of toxins was placed on ceramic rings where tadpoles were then placed. The control groups, Group 1, for the study, contained no rings with toxins. It is stated that the rings do not have any effect on fitness levels or reproduction rates to provide justification and reason for the use of the rings in this study. In experiment 2, quote, all mortality was due to cannibalism. When the toxin is present, it appears as though they almost choose to be cannibalistic. Then the prey on native species preferring tadpole eggs with the toxin than native species over 98.9% of the time, as shown in the table in the bottom right. In experiment 3, and the table listed in the top left, the data does not appear to be significant enough to justify that the toxin causes the cane toads to become more cannibalistic. What it does show is that Armorina was eaten more when there was no toxin present and was eaten less when the toxin was present, which contradicts all other data previously gathered. A possible error that could have caused this result was stated in the results as, quote, predation rates may be attributable to the greater toxin concentrations in this treatment. End quote. This may be due to habitat and ecosystem resource competition from overabundance of cane toads in too small an area with limited resources, but it is more likely that this is caused by the toxin at Bufidamina.